Okay friends, so now we are starting with a new chapter, and that chapter is chapter 5, which is cell structure. First of all, let us see the introduction and the topics that we are going to cover in this chapter. Firstly, we are going to study what all the cell is about, and you already have studied these things in your previous standards. So just let us have a recap of that. As we know that cell is the structural and functional unit of living organisms. So that is what how we define cell, that is a structural and functional unit of living organism. Now as we know, depending on the number of the cell, two type of organisms are there, which is unicellular, the one in which the body is made up of single cell, and the other is, you must be knowing, that is multicellular, that the one in which the body of an organism is made up of many cells. If we take to the example of the unicellular organism, then the examples of unicellular are amoeba, paramecium, yeast, bacteria, and chlamydomonas. There are many more, but right now to you these 5 examples are given to you: amoeba, paramecium, yeast, bacteria, and chlamydomonas. Further, if we just see to their structure, then whether it is unicellular or multicellular organism, the life of both the organism will start from the single cell, that is the first cell of the body, which is called zygote. So the life of the organism starts with the single cell zygote. Now this cell will undergo continuous mitotic division, and as we know the mitosis, that is one cell will divide into 2, 2 to 4, 4 to 8, 8 to 16, and so on. So in this way, from single cell zygote, by continuous mitotic division, a large number of cells will be produced. This group of cells, they will differentiate to form tissues. Tissues will differentiate to form organs. Organs will differentiate to form organ system. So in this way, the entire differentiation is taking place, and finally, when all this organ system combines, there is an entire organism taking place. Now, as we said that the life of the organism, it starts with the single cell zygote. That means even if I select, for example, 10 raised to 4th cell, from zygote it has divided, it has reached to the 10 raised to 4th cell. Now if I select that cell, do you think it has direct contact with the zygote? No, it does not have. Yet the genetic constitution of that cell will be same as that of the zygote. Why? Because at the end it has been originated from zygote. The 10 raised to 4th cell, which has originated from the parent cell, that would be the one which is originated from their parent cell, and when you see to the backward chain, at the end of that backward chain there will be zygote. So whatever number of cells are in a multicellular organism, each and every cell have genetic constitution same as zygote, and that is why we can also say that they have the capability to give rise to a new individual. And that characteristic of a particular cell which has a capability of giving rise to a new individual, that is called as totipotency. So this is the introductory part of the chapter. Now let us see the topics which we are going to cover in this chapter. Now basically, as the chapter states, it says that the cell structure, that means we will be studying all the concepts which are related to the cell. We will be studying the cell, what is cell, cell theory. Then we will be studying an overview of cell basically. Then we will study the type of the cell, which is prokaryotic cell, and the third is eukaryotic cell. In prokaryotic cell, we will be studying their structure, what type of cytoplasm they have, what type of layers are surrounding the cytoplasm, what type of protective structures they have, everything. And in eukaryotic cell, again the same thing, what type of enveloping structures they have, what type of cytoplasm, what type of organelles are present in them, and all the organelles will be studying in detail, which is endoplasmic reticulum, Golgi body, lysosomes, vacuoles, mitochondria, plastids. Then we will be studying cytoskeleton, ribosomes, cilia and flagella, then centrosome and centriole, nucleus, and the genetic material which is stored in them, that is in the form of chromosomes. And finally, microbody. These are the structures which we are going to study in this chapter. And yes, along with that, we are also going to study the membrane surrounding them, which is cell membrane or plasma membrane and cell wall. So let us start with each of the topic in detail.